the other one. Yeah. Is it okay?
I think so.
Okay. Yeah, I'll be here. So this presentation is about the monitoring programs, ecology, and phenology of fall armyworm, and the economic impact of the pest in 2021 outbreak in Kentucky, pastures and soybeans, some insecticide efficacy tests in potted plants and laboratory bioassays, and the massive attack of natural enemies to the true armyworm in forage crops. Bioassay we did with the release of fall armyworm larvae both in 2023 and 2024, and some preliminary evaluation of resistance of a wild population captured on wheat close to Princeton, and the summary. So I'm from Brazil. I've been here for 6 months now, and I was always talking to everybody. The pests in Brazil are a lot challenging because we don't have this cold winter like you have here, and the pests are there all the time, you know, and they just switch from crop to crops, and it's a big challenge. Like here is like, as far as I know, we have like a reset of the system in the winter, like all the pests, like they died with these freezing temperatures. And what happened is like, usually they came from the south, both south of Florida and Texas, and also from Mexico. And this is for most of the pests, and so is for the fall armyworm. So basically what they do, they follow these 2 ways. They came from Texas and Florida and go up there. And what they are noticing in the last few years is they are also doing the way back to overwinter in the south. So we have this migration from south up to north, and that's when we got some populations here. So the problems we're going to have in the season, it depends a lot of the weather conditions, because this is what helps just both bring the pests and also the survival of them in the fields. We are going to talk about this. We are always going to be back and to what I said before, because it's always correlated. You know. So one work that has been done at the station over the past few years, and this is a constantly work, is the monitoring using traps. These are pheromone traps. We have here, this one is a Texas modified cone trap. So we have here a pheromone in this pet, so it attracts the males of the moth, you know, the adults. And then here is the pheromone being released. So the moth come here and they go up, because this is the natural behavior, and then they stay here and they can come back. So they are all here. And another trap we have, it is this bucket trap. They have also a pheromone inside, a lure, to attract the males, and also insecticides. So they get inside and they are poisoned, and then they die there, and that's how we can be monitoring the arriving of these pests. So we place these traps in the borders of the field, and the idea is check when the pests are arriving. So when we start to have males, is recommended like collecting adults in these traps. After that, we are going to have eggs in the field, and than the caterpillars. So this is another kind of trap we used this year to collect eggs. So the females came here in these structures, and they laid the eggs here. And these are one example of an egg mass that was there in that trap. So, and what we saw here is like, this is the number of adults that is collected, the average number in each one of these traps. This is for the adults. And these are the number of eggs collected in these other kind of traps, And as you can see here, when you have several, peaks of collecting adults, we start also to have some eggs in the field. So this was placed on corn, and this one to monitor eggs, we started to do in August because of labor. We didn't have enough people to be doing this work, so we started that a little bit later. But we can get some correlation from when we are getting the adults in the field, and right after that we are having some eggs. So and here, so it was in the corn field, and these other ones was in alfalfa and wheat. We planted later, so yeah, we placed these kind of traps in a cornfield also, but we didn't have any eggs laid there, just in the alfalfa field. As I said, we placed these traps there in August, so it's kind of a little late for corn, and that's basically, I believe, is why we didn't get any eggs over there. So, and by this time of the year, the moths are looking for the crops that are fresh in this time of the year, which is alfalfa and wheat. And all this data you can access in this page here. So you can follow. This is weekly updated, so we can get in real time how is the movement of the pest. I believe you have these in the handouts that they provide you with the presentation. And you come over here in the page, and you have here the pests, So if you click in For Armyworm here, it's going to open these graphs. So, and then you can check when you are starting to have the moth here. These traps are placed in Princeton, but it can tell a little bit about, to have at least an idea when the moths are arriving in the state. So, and this is from 2012. This black line is the year we have an outbreak over there. This is in Princeton, and this is in Lexington. So Lexington from 2024, and in Princeton we have data from 2010, 2012, and 2024. So in 2010 and 2012, we have outbreaks, And as you can see here, we have a high number of moths also, a lot more than the other years. So as you can see, this monitoring work can tell you a lot if we are going to have a bad year with a lot of problems and risk for outbreaks in your crops. So this is based on the IPM, Integrated Pest Management. Like, we need to be doing the monitoring, and after that, recognizing properly the pests and checking how many pests we have in the field to take a decision if we are going to spray something or not. So one important step is to recognize the species we have, and these ones are important on pastures. So this is, we have here the black cutworms. true armyworms, the yellow-striped armyworm, and the foul armyworm. So according to what my supervisor was saying, this yellow-striped, so it's increasing in numbers and importance and damages over the past few years. And the foul armyworm, we always have, and it's always a big issue, like especially in corn and pastures and grass. So these are basically, most of them are really polyphagous, so it feeds in a lot of species. To recognize each one of those, like in the traps we collect adults, you can see here it's really hard to differentiate sometimes, especially you see this one, and the ornithogalis and the foul armyworm, they are pretty similar. So sometimes it's hard to differentiate the adults, but in the traps we have pheromones that are specific. So we know what we are collecting there when we are using this kind of tool, you know, and then you can have an idea what to expect to find in the field. But once you have larvae, like these black cutworms, they are brownish caterpillars with some black spots on dorsum. And the true armyworms, you need to look in the head. They have this kind of V black stripes here in the head. And the yellow striped and the foul armyworm, they have these white stripes in the head. But then the yellow striped armyworm, they have pretty clear these yellow stripes on dorsum, and also, I don't know if you can see from there, some black triangles here. You see that? And you know, and this is something you can differentiate from the foul armyworm. So, and the foul armyworm from other species, like the caterpillars, is they are pretty unique. So you have this Y pattern here. If you see here, this is the pronotum, is the first segment of the abdomen. And if you see, of the thorax, sorry. And if you see here, it has this yellow, this white stripe here that came straight here, and it makes like a upside down Y, you know. And you don't see this here. So it's just more like a V than a Y. The caterpillars, when you see in the field, they don't have that, the head outside like this. So these marks here are pretty much right here when you see them in the field. So the Y is really clear. And another thing is these 4 black dots in the end of the abdomen that is on dorsum. Can you see here? They form like a square. They are the same distance from each one. So in the last segments of the abdomen on dorsum, you can check these 4 black dots like a square. Okay, and how long it's going to take to the pest develop? As I was saying, a lot here, if we are going to have problems or not with insects, it depends a lot of the weather. So both to bring the pests to the state and also for how many generations and how much damage they are going to be able to do that, it depends on the conditions. So the eggs can take from 2 to 10 days, and the larvae from 7 to 22 days, and pupa 10 to 14 days, and adults 11 to 14 days. So when you have favorable conditions, so you're going to have the whole cycle in 30 days. You have unfavorable conditions, so it's 60 days. So we have bigger problems here because we're going to have more moth, and they're going to lay more eggs in the fields, and they have more generations in the same season. So the favorable conditions is years where we have higher temperatures and rain, we're going to see that not that much rain, but and unfavorable conditions are years where we have lower temperatures and drought. So in 2021, we had an early outbreak affecting pastures and soybean in mid-July. This is the kind of damage that the fall armyworm makes on corn. It damages the apical meristem of the plant, so it basically kills the plant, because when you don't have like the point that the plant is growing, so it's all gone. And you see here like those black dots I told you about, how to recognize, and see the Y here, like it's clearly. So the economic impacts, so in 2021, these outbreaks you have early in the season, so it's estimated cost 5.2 million in the pastures, and around 100,000 acres were affected, and half of them needed to be sprayed to control the outbreaks. So again, we have here the moth and then the eggs. These are the first instar caterpillars. You see them really young. They have this black head and greenish body and a lot of black dots on dorsum. So, and later we have these big ones. So they feed a lot and they damage a lot of the plants. So they lay the eggs. We have insects that invest on parental care, so they lay fewer eggs, they have fewer, a smaller offspring, but then they kind of take care of them for a little while until they grow a little bit. And we have other kind of insects that invest in numbers, so they lay a bunch of eggs, so they can afford to be eaten because they have a lot of predation in the field and parasitism and everything. So Spodoptera is one of those that lay a lot of eggs. So we saw those data about the number of egg masses we have. So each one of those have hundreds of or thousands of eggs. And they lay the eggs like this in layers because they have parasitoids of eggs. So the lower layer is kind of protected from these damages. So they save some offspring, which is still a lot. So this is the results of the outbreaks in 2021. If you see here, all this part here damaged, and also these. So they basically came from the border to the center of the field. The insects arrive in the border, so they destroyed everything. And if we are not monitoring, we can face this kind of situation, from one day to another. This is why it's important to be aware when the moths are arriving, and then we need to be pay more attention in our fields to check for, eggs and small caterpillars, because this is really important also for the efficacy of control. We're going to see that later. So this is, again, the eggs. And this is to show you that you saw those traps, the black traps we installed to collect eggs. They are from a plastic fabric. I don't know if I can say that. But anyway, they lay eggs everywhere. And this is basically a behavior they are fall armyworms, so they migrate as larvae in the field. So they most lay the eggs in the borders, so in the poles of the fence and plastic stuff. I have a friend who worked on Spodoptera frugiperda to check the oviposition preference between soybean, sorghum, millet, and corn. And he told me that the results is like they prefer to lay eggs on the walls of the greenhouse, you know. They are really polyphagous. They eat everything, you know, not the plastic, but. Yeah. So again, these are first instar larvae, and they go as an army to feed on the plant. So that's why we see that much damage really quick, you know. Again, here, just calling for attention of the Y pattern, you know. So these are, again, those fields, bigger. So you see here damage. This is a soybean field, and there is no leaves here, you know, no leaves. So they're really voracious. We're going to see more of that. Wow. Escape. Okay. Okay. And this is a work that was done measuring the occurrence of fall armyworm in fields that were sprayed with besiege and unsprayed. So in the fields that were sprayed, we basically didn't find anything, neither one day after spray and a little bit here in the soybean inside the field, 20 days after spraying. And where we have the control, unsprayed fields, we found caterpillars both in the border and inside the field, you know. And these are the number of caterpillars in 20 sweep nets. Are you all familiar with the sweep net? Is, you know, we use to collect the caterpillars to make the samples. So this is, we have results that we are having a good level of control of this product against the fall armyworm. So by the way, this was, we had also an alfalfa field, so that was sprayed, and we didn't find anything there. So then we had the moth. We recognized the caterpillars. We saw how bad they can damage the plants, how we control them when we have high populations. So to provide this information, we tested several insecticides to check what are the best options in the years and environmental conditions. We have outbreaks, so we need to have efficient tools to manage the pests. So we tested Dimethoate, Steward, Besiege, and Warrior. This was a work in alfalfa plants. They were grown outdoors in pots, and also we have another bioassays in laboratory conditions. We're going to have both of them. So the first time the team made this work, they pick up the plants and they isolated the whole branch inside a net bag, and they put the caterpillars inside. So first time they put 10 fall armyworms per branch, and in the second time they put 1. And then they closed that inside of a net bag. So they sprayed the plants, they put the caterpillars, and they closed the bags, okay? So here is some results. This is the plant, the branches from control, and Dimethoate, and Steward, Besiege, and Warrior. as you can see here, you have a lot of damage in the control, almost no leaves anymore, and a lot of damage also in dimethoate, and less in steward, besiege, and warrior. And you see that, like, it impacts the defoliation. So why they did that twice, and they put 10 caterpillars at the first time and just 1 caterpillar in the second time? Because the fall armyworm, they are cannibalist, and they eat each other. So, and then especially when you see here in the control where they have no leaves, they, we have a lot of missing caterpillars. So they did that again with 1 larvae, and you still see a lot of missing caterpillars here in the control, but that's why when they finish the plants, we have no leaves anymore, and as I told you, they eat everything, so they made holes in the bags and they escape. But here they found a lot of dead caterpillars, so if they die, they can't escape. So the products that really work well to kill the caterpillars, we didn't miss anything when you have 1 larva. Here we can have both them escaping or eating each other, Again, steward, besiege, and warrior with more than 90% mortality of the caterpillars in each one. A good product to be registered to control a pest in a crop needs to have at least 80% of control. So these are working pretty good. Dimethoate, not that good, and we saw that they eat a lot of the plants that were inside the bag. So they were making holes in the bags and escaping, so they need another round of these same treatments inside plastic cups, so they can't feed on that stuff. And we see here the control, how they ate a lot of the plants, they basically finished, and this is dimethoate again, so they cause a lot of damage also, and steward, besiege, and warrior. So, steward, warrior, and besiege, we have a good control and still have a lot of leaves. So this is another work to show also the control of the same products. And you see here, like, this is over time, and we have like dimethoate pretty close to the control where we didn't spray the plant, so it's not being that effective to control the foul armyworm. And we see here the besiege and the steward on top of that, like from the first, second day of evaluation, it's basically 100% mortality. And warrior, it takes a little bit longer, but also get some good levels of control. And we see the same here, like 2, 4, and 7 days after application with 1 larvae. So these are the results of mortality. So as we had these good results, a lot of damage in the plants, in the cages, we saw that, we wanted to check that in the field. So we had a plan to release these caterpillars in the field and do the same kind of work, quantify the damage and also spray some plots and see if we can control the caterpillars, and we are going to have, we are not going to lose yield because we are spraying good products. So we tried to replicate the same study we did in cages and in the lab in the field. So as we were discussing before, the environmental conditions are really important to see if we are going to have a big issue or not. We did a lot of work to get that. We released 1,000 caterpillars in each plot. So we have 2 of these growth chambers full of caterpillars. We kept them for 2 weeks until they grow a little bit, and then we released them in the field. But so this is how many cups we have, and we did that 3 times this year. So, but this year, so this is the releasing in the field, a big team doing that, releasing all the caterpillars. But this year, we have the Hurricane Helene, and it brought a lot of rain. So from the day we did this first release, it rained for a week without stopping, almost 24 hours a day, 7 days a week. So we didn't find anything. So we released it again a second time, like 2 weeks later, and again we didn't find anything because after the rain it came a really cold weather. So we were not lucky this year to perform this assay, and we released it again 1/4 time and a third time, and we didn't get that many caterpillars. We released 1,000 of caterpillars in each plot, and we could recover only 2, 1, and most of the plots none, But this is something else. It's both the weather. So we have a lot of rain there, and also the presence of natural enemies. We are going to see that also. And the Hurricane Helene that came in September, so it kills a lot. So, and after that we had the drop of the temperatures. So we didn't succeed on this infestation in field conditions. Part of that because of these weather conditions, but another thing, we went there in the field to check for the caterpillars later, and we were seeing a lot of natural enemies in the field. So this is a work that was conducted in 2023. Then they quantified the natural enemies that were happening there. So these are, again, the traps, the moth, and then they have the outbreaks again. But okay, this work is basically to show that how many natural enemies we have there also in our fields. So if you see here is eggs of parasitoids, and these are larvae coming out, and they are after that pupate, and this is carabid larvae, and these are cocoons of parasitoids. So they found a lot of eggs of parasitoids and also larvae of carabids. So we have a lot of beneficial insects in the fields also. So these are all the species that were found, and this is published. So this is in forages, like 3 fields. And if you see here, so in clover, crabgrass, and taft grass, so we have a lot of larvae, more in crabgrass than taft grass. You see here like 11 larvae per square meter, 33 larvae, and less in clover. So, but we have these big issues in the crops we are growing because monoculture. When we have a whole field of one crop, everything we are going to have there is something that is going to feed on those plants, right? And it's the same for natural enemies. When we have a lot of pests, so this is food for natural enemies, so we have also more parasitism, more mortality in these fields. We have more insects. So this is something else that we need to be aware of and keep monitoring, because not all the times we are going to have the pests. We need to spray, because if we are going to have good numbers of natural enemies, and also we have like the weather conditions I told you, like we released 1,000 of caterpillars in 10 or 15 feet plots, and we didn't recover almost anything because we have a lot of environmental conditions and natural enemies that were killing the pests. So this is why it's important to be monitoring, This is wheat field. We have there close to Princeton. I don't remember the name of the county, but it's like 1 hour from Princeton. And we have an outbreak. This is September, October of this year. And the farmer basically lost everything. I went to this field, and the whole field was like that, And this is a field that he had sprayed the day before we went there. So, and we found a lot of caterpillars. And he sprayed a pyrethroid, and was not working. I'm going to tell you more about that. So I collected some caterpillars of this field and took to the lab to see if they were going to still die, because he had sprayed the day before. So, but even like 1 day, 3 days later, like from the 80 caterpillars I brought, like 15% died, So it was not working. So he sprayed generic beta-cifultrin. Then we pick up these caterpillars here, and we rear them in the lab, and we had a colony. So we tested another products, again, to find an efficient tool to control these caterpillars. I was there in this property, and I was talking to the farmer. So one thing that is important to mention, he used a product, this beta-cifultrin. He was using the whole season in soybean to control stink bugs. It's pretty effective against the stink bugs, but it's not that good for caterpillars. And then when you don't kill everything, you have the development of a resistant population comes from a lack of control. So a few individuals that have some characteristics that are going to confer them some resistance, they are not going to die, either because the product is not penetrating well, or because they are degrading, enzymatic degrading the products, or because they have a mutation on the target site of the insect, so the product doesn't work. So either one of these traits are going to go to the offspring, so we're going to have a lot more individuals in the next generation. And when you use the same product over and over and over, we are just doing this selection process to have, in some point, we are going to have a whole population that is resistant to the, to this active ingredient, and we are not going to kill anything. So he was probably these caterpillars. They were there in the soybean in low numbers, and then he came using that for stink bugs over and over. So when he planted the wheat, so small plants, he had a lot of damage. So, and he lost the whole field. Okay, one thing is important is the timing to control that, because as you can see here, we have good products like if you check the Baytroid is the product that he was spraying there, is the beta-cifultrin, and it has lower mortality than X-Rail and Mustang, okay, the other products. When you change the product, you can kill the resistant population. But then it's really important also the timing for application. You see here that the smaller caterpillars, they had almost 90% mortality. And when you went to fourth insta caterpillar, like it was around 70, even the good products. So imagine that in a fifth insta caterpillar, 6. So again, adults, eggs, small caterpillars. So we need to be aware of all of that, because if we miss the time to spray, even a good product is not going to work, you know. So I think that's it, what I had to present. Basically, the monitoring is a helpful tool to prevent losses from fall armyworm outbreaks. If we keep an eye of those data, we see when the moths are arriving. So if we have good numbers of adults, we better start to monitoring our fields, because as soon as we detect caterpillars in our field, the earlier we can detect that, the earlier we can spray that, the smaller the caterpillars are, the better is going to be the control, okay? So in 2021 and 2024 bioassays, the insecticides Besiege was effective to control fall armyworm, and so was Steward, Mustang, and Warrior. Baytroid and dimethoate did not control the fall armyworm in potted and laboratory bioassays. So dimethoate was a laboratory colony, was not even this one. So the bioassays we have with the Baytroid came from this field that was, again, the caterpillars was being exposed to the same active ingredient for a long time, so maybe that can contribute to the loss of efficacy of this product. Okay? So, and this was also the first study on the true armyworm that reported the occurrence of these parasitoids in forages. So the armyworms in teff grass and crabgrass had higher occurrence of parasitoids than in clover. And we didn't conduct yet studies with the LD50s and LD90, the lethal doses to kill 50% of the population and the 90% of the population for the fall armyworm population collected in that wheat field. But the preliminary results we have indicate that this population had developed the resistance to Baytroid. So that's it, I think. Thank you, and if you have any question,